Welcome to Rappaport Connects, where we interview notable Rappaport Foundation alumni and fellows. My name is Sean Horrigan. Today we're joined by Dr. Rudy Tanzi. Dr. Tanzi is a world-leading neuroscientist and geneticist specializing in Alzheimer's disease and brain aging. Dr. Tanzi is the director of the Genetics and Aging Research Unit and the director of the McCance Center for Brain Health at Massachusetts General Hospital. He serves as the Joseph P. and Rose F. Kennedy Professor of Neurology at Harvard Medical School. Dr. Tanzi discovered numerous Alzheimer genes, including the first three, and directs the Cure Alzheimer's Fund Genome Project. Dr. Tanzi has won the highest awards in his field and is a member of the National Academy of Medicine and is one of the top 1% highly cited researchers in the world. He has co-authored over 750 publications with over 180,000 citations and is a New York Times bestselling author of several books. We are so honored to have him joining us here today. We had the pleasure of Dr. Tanzi speaking at a Rappaport Foundation board meeting a couple months ago, and the information you shared was so profound that I wanted to share it with a wider audience. So, Dr. Tanzi, thank you so much for being here today.
Oh, you're welcome. Thank you for having me, and especially thank you for the support from the foundation. It's unbelievable.
Why don't you share your history with the founding of Cure Alzheimer's and your connection with the foundation and the genesis around all of that?
Sure. So I've been in the MassGeneral Harvard system my entire career, from student to professor. So when I first started here, I worked on the first project that ever found a disease gene using genetics. It was for a movement disorder called Huntington's disease. And I started studying Alzheimer's disease in the early 80s and found the first gene, the amyloid gene, and never turned back in terms of finding Alzheimer's genes, figuring out how they work, making model systems, and then coming up with drugs. And one of the drugs that I invented is now considered the lead drug in the world for Alzheimer's in terms of the hopes for having something to prevent the disease. It's called a gamma-secretase modulator, where Phyllis and her supporter, Cure Alzheimer's Fund, really helped make that possible. So we have a long history here. Cure Alzheimer's Fund began over 20 years ago when I met with Phyllis first after a donor event at Mass General and explained to her that there was this new technology coming out around 2005 where you could scan the entire human genome and find all the disease genes at once for a certain disease. Before that, we had a peck and paw, one at a time, just one at a time. And she was thrilled by that. And she said, I'd like to help support that. Next thing you know, we had 3 founding families. We said, let's start a 501c3 to fund this, and who knows where else it will go. And that was called the Alzheimer's Disease Research Foundation. And then Phyllis, being Phyllis, said, I don't like that name. It's very boring. So let's do something more fun. So she came up with Cure Alzheimer's Fund, which is our name today. And those 3 founding families, including Phyllis and her foundation, and Henry McCance and his foundation, and Jeff Morby unfortunately passed away, but Jackie Morby still supporting, and other families now. It's unbelievable that this little 501c3 20 years ago now is giving out $30 million a year and is one of the major funders of the highest impact Alzheimer's research in the world. And I'll just finally say that everything we're learning preclinically, translational medicine from the Genetics and Aging Research Unit that I direct at Mass General, 11 labs, 70 people, Alzheimer's research. Now we have the McCance Center where we can do the clinical trials. And now we have the generous support of the Rappaport Foundation to fund one of those trials in its entirety. So we're thrilled about taking as many shots on goal, and we're thrilled that the Rappaport Foundation is allowing us to take a really big shot.
How much has Cure Alzheimer's raised? Is it $250 million in its 20-year span?
That sounds about right. It might be even more.
Yeah.
And the really cool thing about Cure Alzheimer's Fund is that when someone from the outside, anybody gives $1, it's $1 out. Most other disease foundations, $1 in might mean 20 cents out or even less. But Phyllis and the other families and board members cover the overhead. So it's very unique that $1 in is $1 out.
It's a very impressive model. I'm always promoting it to people who are passionate about Alzheimer's. Rudy, you discovered the concept of Alzheimer's in a dish. And how did that change your research? And tell us a little bit about that concept.
Yeah, so up until 2014, there were 2 big gaps in the field. One is more technical. We didn't know whether the amyloid plaques of Alzheimer's disease caused the other main pathology, the tangles that choke the neurons from inside. Because when you would put the Alzheimer's genes that we discovered that create more amyloid plaque into mice, they'd make amyloid, but they wouldn't get tangles. So the field was saying, does amyloid really cause this disease? As we would learn later, mice can't make tangles. That's why they didn't have them. So the debate was about nothing. But then there was still a question, how are we going to show that amyloid creates tangles? Like that one pathology drives the other? Because we know from brain scans, on average 20 to 25 years before you have symptoms of this disease, the amyloid plaque begins accruing in the brain. So the question was, does that drive the tangles that really kill the nerve cells? And eventually the inflammation that kills 10 times more nerve cells in the brain. So Do-Yeon Kim in our group, who's also been supported by Cure Alzheimer's Fund, is also an affiliate member of the McCance Center. He and I worked out a way to create a mini human brain organoid the size of a piece of rice, like a little piece of gel, because your brain is gelatinous. And we were able to put the Alzheimer genes in and create human neurons, human glial cells, all the different cells of the brain in a gel matrix. It's magical. You put them in and they know what to do. The nerve cells start talking to each other and firing. The glial cells start housekeeping and pruning the neural network, the other cells. So like you don't have to do anything. Just put them together in gel that mimics the brain and they go. And then you put the Alzheimer genes in and now you start making amyloid plaque. So I remember Do-Yeon Kim came into my office about 4 weeks after he built the first organoid. And he said, we have amyloid in the dish. I said, great. How about the tangles? He said, I'll be back. and literally he said that. And literally 2 or 3 weeks later, he comes back and he says, we have tangles. I said, great. Now see if you block the amyloid with one of our drugs that we've made. Do you stop the tangles? He came back. He said, yep. You stop the amyloid, you don't get the tangles. That was the first proof of concept of how the disease begins. Amyloid causing the tangles, the cell death, and then eventually inflammation in the brain cause, everything goes awry. I like to say amyloid is the match. It's lit by genetics or it can be lit by infection or pollution or microplastics. There's many ways to light that match to get amyloid. That match lights the brush fires of tangles. I call them brush fires because they spread. The tangles propagate and spread like brush fires. Then eventually that causes the wildfire of neuroinflammation. Then you get the disease. So if you want to nip it in the bud, you have to hit the plaques and tangles. So how do we find drugs that will do that? Can we repurpose drugs to do that? Can we repurpose natural products to do that? I also have new drugs I'm making that do that Cure Alzheimer's Fund helped with. 25 years in the works, finally going into clinical trials now. But here's the problem. Every time you had an idea for a drug, you had to put it in an Alzheimer's mouse that wasn't a complete model of the disease. You had to wait a year, one drug at a time. Now with the dish, Alzheimer's in a dish, picture an index card sized tray with 96 wells in it. And each one you have a mini human brain organoid where the whole Alzheimer's process is taking place. What takes 20 to 25 years in a human brain in 6 weeks in a dish. It made discovery of drugs and screening for drugs 100 times faster and 100 times cheaper than when we had to do one at a time in a mouse for a year. Now you're doing 100 or even hundreds at a time in 6 weeks. So that's why when this discovery came out, people got that right away. And, the field responded and the media responded. I got on the Time 100 most influential list that year because of this discovery. We got the Smithsonian Ingenuity Award that year, the highest award for invention. And I'm not saying this to brag. I'm saying it to say that even the public got how big this discovery was. Cure Alzheimer's Fund, Phyllis Rappaport, the foundation made this possible. Took a chance on a big idea, high risk, high reward. And now as a result, here we are with all these combinations of known safe drugs on the shelf right now, natural products on the shelf right now that can be combined to hit all these pathological steps of Alzheimer's. And all we have to do is the trials. And at the McCance Center, we're set up to do those trials. We just needed the funds. And this is where the Rappaport Foundation just stepped in and gave us the funds to do an entire trial on one of our combinations. And ultimately, I figure if we can do a dozen different combinations of these repurposed drugs and natural products that stop all the disease steps of Alzheimer's, we're going to hit one or two. And then as soon as we have a combination of safe, affordable, known drugs or natural products, we can democratize early detection of the disease by a blood test, followed by early intervention, nip it in the bud, eradicate this disease. The dream is starting to come true now.
It's so encouraging. As someone personally whose family has been very affected by Alzheimer's, this is just a light at the end of the tunnel. Which of the compounds you're currently working on show the most promise and are the most encouraging to you?
So let me just tell you the buckets we have, right? We have compounds that stop the amyloid from forming. Second bucket, it stops the amyloid from causing the tangles. Third bucket is getting cells in the brain called microglia, the housekeepers, to eat the amyloid and get rid of it. In fact, the two newly approved Alzheimer drugs, Leqembi and Kisunla, that's what they do. They prompt the microglial cells to eat the amyloid. That's how to get rid of the amyloid. But they're very expensive and not very safe. Much better to have a little white pill that's safe and affordable to do that. That's what we're going after. And then the fourth bucket is those same microglial cells that eat the amyloid. If they get exhausted or overwhelmed, they go from housekeeper to killer. They're programmed that if they can't work anymore and that they think that part of the brain is toast, they just destroy it. That's inflammation. That's what causes the bulk of cell death. So very simply, what we're doing is reprogramming the microglial cells to be better housekeepers, eat more amyloid, like the two new approved Alzheimer drugs do. At the same time, we're telling them to stay chilled out. If they get exhausted, overwhelmed, keep calm, carry on, housekeeping. Don't destroy the place with neuroinflammation. So each combination we're doing is one drug or natural product that says, eat more amyloid, housekeep better. The second one says, along the way, don't get upset, don't cause neuroinflammation, keep going. And the best combination we have, the Rappaport Foundation is funding right now for our first clinical trial, together with funds from Henry McCance and the McCance Foundation, together with a large gift with matching that we received from an anonymous donor. So that was a $20 million matching gift. And thanks to Henry McCance and Phyllis Rappaport, the foundations, and also personally, we're already up to 7 or 8 million of that $20 million match.
Fantastic. How far along are these compounds towards clinical testing? And are they more intended towards prevention, early intervention, or treatment?
So technically, what these compounds will do is stop the pathology from accumulating. Now remember, the pathology of Alzheimer's starts accumulating on average 22 years before symptoms. So we need to know who 20 or 30 years in advance has started the pathological process in the brain. It's going to take all that time for the amyloid and the tangles and the inflammation to finally lead to symptoms. By the time you get to symptoms, you've waited too long. That wildfire is blazing. We have to blow out the match and stomp out the brush fires early. So luckily, we now have a blood test that will tell you if you're making plaques and tangles in your brain right now, even if it's decades before you might have symptoms. So that's the early detection. So if we find safe, affordable combinations of known drugs, natural products, or even my new drug that we invented, the gamma-secretase modulator that Phyllis helped support. Early on in life, let's say, certainly by 50 years old, everyone would have the blood test. Your doc says, yeah, you know what, we're starting to see a little signal here, just like a cholesterol test. Some people early in life say, oh, I have high cholesterol. I'm going to take a statin like Lipitor and bring it down. Or I'm going to change my lifestyle, bring my cholesterol down. Well, here you do both as well. I have a lifestyle plan, but sometimes it's not enough, just like with cholesterol. So you'd have drugs that go in and help turn the amyloid production down. That's my new drug I'm developing. I say new, it's been 25 years of just going into trials. That's the gamma-secretase modulator that my late colleague, Steve Wagner, and I invented. But then these new products, the natural products and drugs we're screening, those combinations will clear the amyloid away by getting the microglial cells to eat it and stop the glial cells from becoming reactive and causing neuroinflammation. So right now, it's going to take a while for my new drug, as so to speak, to get approved. We're just starting the safety trials now. But in the meantime, if you have safe approved drugs on the shelf, you have natural products that are already safe, you can get going right away. So the trials we're doing will basically ask, are we getting the pathology in the brain to go down? How do we know if it's going down in the patient? You do a blood test. The same blood test that says, oh, you're starting to make plaques and tangles at this level, X. Now you treat for 24 weeks with 2 products to get the microglial cells to eat more amyloid and remove it, stop the inflammation. And then you do the blood test along the way for 6 months and say, yeah, your amyloid's going down. Yeah, your tangles are going down. Yeah, your inflammation's going down. Once we know that's working, then we talk to the FDA, or in some cases it's over the counter, and say, get that blood test covered by insurance. Right now, it's not covered by insurance because it's not actionable, meaning if the test is positive, there's nothing you can do about it. So the insurance company say, we won't cover it. But when it becomes actionable, meaning, oh, you had the blood test, you're on your way, but here's a combination of 2 drugs and natural products that will help, and the FDA agrees. Now everyone gets that blood test who's older, at least by 50, and you start helping those people to prevent the disease. So to answer your question, the same drugs that treat the pathology of the disease to reduce it are going to be used early on, after early detection, early intervention, to prevent the disease. That's how we nip the disease in the bud. That's our vision for eradicating Alzheimer's disease.
What do you see as a realistic timeline for all this in terms of rollout?
Well, every trial, even though it's a 6-month trial, you have to recruit, and it's serial recruitment. So each trial would take at least a year. The first one's going right now. And, every time we take a shot on goal, right? Let's try this combination. Does it work? Let's try this combination. If we do 10 combinations, even conservatively, pessimistically, I think we'll have 1 or 2 hits that will do the job, that can safely bring the pathology down early in life after early detection. So maybe this first trial might get us there, maybe the second, maybe the third. But because we're using known, safe, approved drugs, repurposed, or natural products that are repurposed, the road to using them with the FDA is 10 times faster than a new chemical entity that's never been in humans before, like a new drug. So I'm hoping that within the next 3 to 5 years, there's a great chance in my mind that we'll have some safe combination that we can have a talk with the FDA and say, let's make this test available. Let's have insurance cover it. And guess what? You know how many Americans are going to be positive for this test if they have it? Minimally 10 million. Some people estimate as many as 50 million because this disease is so rampant, and it begins in just about everybody decades in advance. Of course, if you have a family history with early onset, you do the test earlier in life. But now you might have millions of people just like that on a safe combination of known drugs or natural products nipping this disease in the bud. You said light at the end of the tunnel. For the first time, we are seeing light at the end of the tunnel. And it's more than a pinpoint, and it's getting pretty bright because of this new approach. Combine that with the new drugs we're developing, again, that Phyllis Rappaport helped make possible with the Cure Alzheimer's Fund. And I envisage this. You have your test. My new drug, called a gamma-secretase modulator, will turn off the tap. Picture a sink overflowing with water as the brain's starting to fill up with amyloid. The drug I'm developing, the new drug, turns off the tap. The drug that gets the microglial cells to eat the amyloid is like opening the drain, rotor-rooter. And then just in case that sink is going to do damage to the house, inflammation, the third part of the formula stops that from happening. So I like to use this kitchen sink analogy. Like by the time we diagnose Alzheimer's, the sink's overflowing with water. The water's getting into the floor. It's hitting the electrical circuits. It's causing fires, inflammation, right? We can't wait that long. We have to keep that sink low, turn off that tap, unclog the sink of it, the brain of amyloid as early as possible. We have the blood test to tell us if we're doing that. And now, thanks to our clinical trials at the McCance Center and the support of the Rappaport Foundation, we're taking our first shot on goal. And we actually have the funds. We've actually raised about 28 million so far. So we're being very careful in terms of what combinations we pick, but we're ready to take those shots on goal right now. As soon as a combination's ready and validated, we start the next trial. And we get the money to do that right now. Not all the trials we want to do. We have enough money to do a few trials, but we're on our way.
Fantastic. What combinations look the most promising at the moment to you?
The first combination we're using is 2 natural products. The natural product that gets the microglial cells to eat the amyloid is a soy derivative. It's called a flavone, but it's not estrogenic. It doesn't cause estrogen to be produced. So it's a safer soy flavone in that regard. And it's called ipraflavone. Ipraflavone is available over the counter here for osteoporosis. So you say, why are you using an osteoporosis supplement for Alzheimer's? Why? Because we were totally agnostic. We had no ideas about what was going to work. We just screened everything. We screened 4,000 natural products, all 1,800 FDA-approved drugs, and said, what works? What stops the amyloid? What prevents the tangles? What clears the amyloid? And ipraflavone came up as the number one, even among drugs. It's an over-the-counter supplement. It came up number one for getting microglial cells to gobble up amyloid. So that's the one that gets them to eat the more amyloid. The compound we're using to keep the microglial cells calm so they don't become inflammatory as they're doing this job of eating up more amyloid is called parthenolide. It's a natural product available in an extract from a tree. It's called feverfew. It's sold as a supplement mainly for migraines. In fact, especially pediatric migraines, because it's safe. And if you reduce inflammation in the brain, you can treat migraines. So that's the second one. And now the beauty is the ipraflavone helps the feverfew parthenolide work better. So the two together work additively, to some extent, even synergistically. So that's the first combination. In the trial, we got IND approval from an FDA. We got IRB approval. We're off and running. We're recruiting patients now. It's begun. First shot on goal, stick on puck.
Yeah.
or
I like your analogies. Sinks, hockey, and wildfires are all very good. And
well, what was it? Wayne Gretzky said, I never scored a goal on a shot I didn't take. So we got to take shots.
Yeah. So let's look into the future. Once these drugs are approved, let's say, in that glorious day, would a 40-year-old get tested to see if they're susceptible to Alzheimer's? And how do you envision that? Would they start taking some medication if they showed signs?
At that point, everyone by 50 years old should test. Because if you look at Alzheimer's prevalence in the 70s and knowing it starts 20 to 30 years before, it's like a colonoscopy. You do it after 50 years old. But if you have a family history, I say take the first degree relative who had the earliest symptoms of the disease, take that age, subtract 30 years, and that's when you should get your test. So if you had an uncle or a dad who had the disease symptoms at 70, you're going to take a look at 40 years old. But on average, it's 50. And if the FDA agrees with us that we're preventing the pathology from accumulating, whether it's a new drug or no approved drug combination or natural products or something they're in, they would cover that test. And then you'd start taking these drugs the same way you take a statin for your cholesterol. In the morning, you add a little pill that not only brings your cholesterol down, but brings your amyloid and tangles down, and inflammation. So you start this early in life. Look at what we did for prevalence of heart disease by managing cholesterol. It's perfectly analogous for amyloid and Alzheimer's. If you wait to treat amyloid in a patient with full-blown Alzheimer's disease, that wildfire is still blazing. The tangles, the brush fires are still spreading. What you've done is blow out the matches. That's what Kisunla and Leqembi do. So the patient doesn't really get better with the new approved drugs, but they stay more stable and you don't have new fires starting. But wouldn't it be nice if you could know when those fires are about to start and put them out before there's any damage? There's no brush fires, there's no wildfire yet. Do it at 50 years old. Well, to do that, to democratize it for everyone, whatever you do has to be safe enough to do for decades, and affordable because millions of people are going to need it. That's what we're aiming to do. That's what the foundation is allowing us to do.
Well, we are so proud to be funding this incredible research, and it's so important. So thank you. I wanted to end things by sharing a side of you people might not know about. Can you share a little bit about your music and your career? We were talking a little bit about your playing with Aerosmith, and I loved hearing about that. So I bet people would as well.
Well, I've always been a musician, and in college I minored in music, but mostly jazz, piano. My heroes are Bill Evans and Keith Jarrett and a lot of the great jazz pianists. But I also, to make side money, played keyboards in rock bands. And I had to stop when I went to, as soon as I started at Harvard Medical School, that's probably the last time I played with a band. I didn't have time anymore. And ironically, it was science that brought me back, because in 2009, GQ magazine and the Jeffrey Bean Foundation did this campaign called Rock Stars of Science. And they picked scientists who were rock stars in their field to pose with real rock stars. And they had these posters and everything campaign. So Francis Collins and I represented Alzheimer's and genetics. Francis Collins did the Human Genome Project. They had Tony Fauci for AIDS. They had the Nobel Prize winner Harold Varmus for cancer. So now Francis and I were with Joe Perry for the photo shoot. And Francis plays folk guitar, Bob Dylan stuff, and sings. And we get done with the shoot, and Francis said to Joe Perry, the lead guitarist, founder of Aerosmith, Rudy and I are both musicians. We should play sometime. I'm like, oh man. And Joe, being a nice guy, said, sure. All right, let's do some like Alzheimer's awareness thing in DC together. So we did that. It was really a lot of fun. We played a hotel, we played for Congress. And when we got done, Joe Perry said to me, hey, you should come and jam with the band. I went to jam with the band. They said, hey, try this song. And it was a cover of a Bob Dylan song that they were going to play on the Jay Leno show. And I played the organ to it. And said, we need a keyboard player for this. You want to come and play this with us on Jay Leno, the Tonight Show? Meanwhile, we're rehearsing in the Aerosmith studio where they're doing their new album. And Steven Tyler and Joe Perry said, hey, we should put this guy on the album. And they said, you want to play on the album? I said, sure. So next thing you know, I'm playing on the Aerosmith album. And then ever since then, I've remained as a studio keyboard player for them. And I did Joe Perry's last 2 albums, all of the keyboards for that. And I still doing the studio keyboards for folks in that whole satellite today. But now it's not so glorious. Now I just do them from my home studio on Zoom. So it's not as much fun. But,
I bet.
But on the other hand, I don't have to travel and I can do it from home. So that's good.
Right. Too. Aerosmith, I grew up, they were in my neighborhood in Somerville growing up. So I would see them from time to time. And Joe Perry, they're really nice guys. And
oh, they're great guys. All of them. Tom Hamilton, they're all great guys. And now on my own music, I still do jazz piano compositions. And so I have my own website. It's all just piano solo or trio stuff. n1m.com/Rudy Tanzi. And I have probably 5 hours of original piano compositions and trio pieces on there that are more along the lines of what I was trained to do, more Keith Jarrett, Bill Evans style. Not that I could ever play like them, but that's what I aspire to try to do. And it's interesting because on that website, I get 25 to 30,000 listens per day. Like more people are hearing my music every day than reading my scientific papers. That's okay.
Yeah. Well, I think that's going to change pretty soon.
Oh, no. It's I'm scientific. We had our unit, the Genetics and Aging Research Unit, where we do the preclinical work that leads to the trials. Thanks to Cure Alzheimer's Fund, we co-authored 52 papers last year in our unit. That's 1 per week. That's what this funding's allowed us to do. And then because we got support to do the trials, including from the Rappaport Foundation, it's seamless. We can go right from the lab here in Charlestown over to Assembly Row in Somerville and start a trial on whatever looks promising. And that's exactly what we just did with the first funding from the Rappaport Foundation and from Henry McCance.
A rock star in the studio and in the lab, Dr. Rudy Tanzi. I had to say that.
Oh, jeez.
Thank you so much for all you do and for taking some time to share. The way you teach us about what's happening is so approachable and understandable. So thank you for that. And it's a very encouraging time.
Well, thanks for having me. Thanks for your time. And thanks to the Rappaport Foundation for supporting our work. The light at the end of the tunnel is getting brighter. We hope it keeps getting brighter with your help.
Take care.
Thank you.